talking about the contractile protein here, this is formed by G-actin. So G-actin has the binding site for myosin. Because these two proteins will eventually come into contact, there needs to be a site on actin for myosin to actually adhere and to bind onto this actin. So if you look at one of the monomers of actin, this is called G-actin. It's G-actin, G standing for globular because it's a round protein or a globular protein. And then many of these G-actins link up together to form F-actin. F standing for fibrous. This is now a fibrous protein because it forms into this strand. It's kind of elongated fibrous strand. And so we have F-actin, two of those strands wrapping around each other in this double helical arrangement to form actin. Okay, so just to iterate that here again, we have a single globular protein, G-actin, linking up together, kind of like pearls on a string to form F-actin, which is a fibrous protein. And then two of those F-actins wrap themselves around each other, right, kind of like DNA, a helical sort of arrangement. And then that forms our actin monomer, our actin filament. Now, as a part of that actin filament, there are two other regulatory proteins. So these proteins are not contractile. They're not actually involved in the shortening of the sarcomere. They're not actually involved in the actual shortening of the myofibril and the sarcomere, but they're involved in the regulation of muscle contraction, hence the term regulatory proteins. So one of them is called tropomyosin. And tropomyosin is this thread-like regulatory protein. So the purple strand or string here that's wrapping around, it's actually covering up the binding sites for myosin. That is very intentional. So muscle contraction, we made this, this point before, it's really important to be able to turn muscle contraction on, but also just as important to be able to turn muscle contraction off. And so tropomyosin is going to help to regulate this process by covering up those binding sites and preventing actin from binding with myosin. This is how we can regulate muscle contraction and turn muscle contraction off when that time is appropriate, okay? So that is tropomyosin, our thread-like regulatory protein that is temporarily covering up these binding sites, blocking that interaction with myosin until it is time to do so. The second regulatory protein here is troponin. And troponin is actually a complex of three other proteins, that form one troponin complex. One of those complexes here, one of those proteins on this complex attaches to actin, so kind of the deepest one that we can see here. So this pink structure is that troponin complex. The structure that is closely adhered to actin is the troponin adhering protein of this complex. The second protein in the complex, the one in the middle, attaches to tropomyosin. And then the third complex is what binds calcium reversibly, okay? So calcium is also playing a vital role in regulating muscle contraction. So when it is time for the muscle to contract, calcium will be in the sarcoplasm. It will come into contact with the troponin complex, specifically at the protein complex that can bind calcium. And then when it binds that calcium, right, when that troponin binds to calcium, or that calcium rather binds to troponin, then the other protein that is a part of this complex, the one that is attached to tropomyosin, our thread-like regulatory protein, that will help to drag that protein away from the binding sites, which is going to expose those binding sites and allow myosin to reach up and to grab onto those binding sites, okay? So all of these fine details is going to be really important functionally when we talk about muscle contraction. So just to kind of reiterate how these regulatory proteins are acting here again, we've got this tropomyosin protein covering up those binding sites, waiting for calcium to come in. When calcium comes in, it binds to one of the proteins that are a part of this troponin complex. That will cause the entire complex to have this shift, which will drag the other complex or the other protein that is a part of this complex, which will drag that tropomyosin off of those binding sites and allow for them to be exposed such that the myosin head can reach up, grab onto those binding sites, and we can start to initiate muscle contraction. Okay. Let's talk about myosin on the other hand. So myosin, which is our thick filament, this is also kind of a helical-like structure. It's basically formed by these golf club-like filamentous strands that wrap around each other. And we have two heads that are forming this thick filament, and it's got two binding sites on it. One is binding the actin. So whereas we saw actin have a binding spot for myosin, myosin also has a binding spot for actin, which are the myosin heads that we're seeing here. It's also got a second site that's going to bind ATP. And so the binding of ATP onto the myosin heads is what will energize this process. So as we mentioned before, myosin is going to get energized by ATP. It's going to reach up via the actin binding site, also via the exposed actin, the exposed myosin sites here on actin. We're going to be able to have an interaction with myosin and actin, and then myosin will drag actin towards the center of our sarcomere, and that'll cause the Z lines to come closer and closer together, which will shorten our structures and eventually bring about back contraction. Okay. So myosin head is facing out towards the I band, right? The tails are in at the M line, the heads are out towards the I band where that actin is found, and then it can reach up, make an interaction with actin and drag it closer to the M line, which is going to shorten that sarcomere. Okay. And just to kind of reiterate here, or just to describe here, these structures, these heads that are sticking up form cross bridges. And we call these cross bridges because these are the regions that are sticking up to find that actin, find that myosin site on the actin and make an interaction with actin, hence forming these bridge-like structures that we call cross bridges. And when we have these interactions being made and broken, that is called cross bridge cycling, which we'll talk about coming up. All right. Another important protein that is found along the sarcomere is titin. And titin is a stretchy, elastic protein. It's going to help support the sarcomere. It's going to help to support these structures here. It's found at the Z line, and then it's connecting all the way to the end of the thick filament. Okay, so it's anchoring between the thick filament and the Z line. So really that anchor between the M line and the Z line. And it allows for that stretchy kind of recoil nature to the sarcomere. So the sarcomere is going to be shortening and shortening and shortening, and we want it to be able to rebound when it's time to relax, okay? So it's going to have that ability to shorten and expand very easily, and that is brought about by the stretchy, elastic nature of titin, which is this protein over here, right? So that is not only providing flexibility for the sarcomere to shorten and relax, but it's also helping to anchor and structurally support that sarcomere as well. All right, I'm going to pause here and see what questions we have. And in the interest of time, we're not going to go over this drawing in real time, but I will explain it. I will also share it in our recording folder if you would like to go over and kind of redraw this, recreate this as a better way to understand this. Okay, so here is what our sarcomere looks like. And just for some context, we're thinking about a muscle body here, wrapped within the endomysium. We think about a muscle fascicle, excuse me, here, wrapped within the perimysium. We're thinking about the muscle fiber here, wrapped within the epimysium. Okay. Well, sorry, I think I flipped that around. So it's endomysium, perimysium, and epimysium. Excuse me. No, I mixed that up. Sorry. Epimysium, perimysium, and endomysium. Epi being the outer covering, endo being the inner covering. Okay. Then we have the myofibril. So the myofibril are these many units that are inside a muscle fiber or cell. These have the repeating units that are the sarcomere, and the sarcomere is what we're looking at. So one of these little units here is what we're zoning in and really focusing on and looking at. So we've got the thick filaments, which are these structures out here in the middle here. These are anchored together by the M line. So this is the M line. This is anchoring the thick filaments together, or the myosin filaments together. We've then got these cross bridges, so they're sticking up, the heads that are sticking up here of the thick filaments of myosin. These are called cross bridges. And then we've got the thin filaments, excuse me. So the thin filaments, which are actin, are the red structures here. These are forming the outer periphery of the sarcomere. These are being joined together or anchored by the Z line. Okay, so the boundaries or the borders of the sarcomere are the Z lines, and these are formed by the thin or actin filaments. Now, as far as our other staining regions, we've got the H zone. The H zone is the region which is just thick filaments. So only thick filaments, no overlap just yet. This is the center of the sarcomere. It's around the M line and is formed by only thick filaments. We've then got the A band, so the area where we have overlap. So you can see that the black structures with the cross bridges are now overlapping with the red structures. That is where we have our A band, a slightly darker region that is formed by overlap between thin and thick filament. We've then got our I band, and I band is representing the area of only thin filament. So the area where we only have these red structures, right up until where we have that boundary between the thick and thin filament. This is called the I band, which is no overlap, only formed by the actin or thin filaments. And so we've got these different zones, so the I band on either side, the Z line being the center of the I band, the A band in the middle, the H band being the center of the A band, the H zone being the center of the A band, and then the M line being the center of the H zone. Okay. The other thing that we have here is titin. So that's this regulatory protein, the stretchy elastic protein that is helping to anchor the M line at the center here to the Z line at the periphery here by the connection to the thick filament. So titin stretches out from the Z line, anchors on to the very end of the thick filament, which is then connecting to the M line. And that kind of structurally supports the entire thing as it is moving back and forth, but it's also giving it some stretchability, some flexibility to contract and relax as this thing shortens and expands. Okay. And so just to kind of reiterate what's happening here when muscle contracts, these cross bridges, which are the heads of the myosin filaments, thick filaments, are going to reach up, they're going to stick out, grab onto the actin, so this red strand here, and drag the actin towards the M line. So as we drag this red structure, which is the actin, towards the M line, these Z lines get closer and closer together. That is how we shorten the sarcomere. And as we shorten that sarcomere, we shorten the myofibril, so the entire thing starts shortening. We then shorten the entire muscle fiber, the muscle cell. We shorten the muscle fascicles, and then we shorten the entire muscle. And when we shorten the muscle, we kind of drag the tendon. So when I lift my biceps, when I do this, that is just the shortening of my biceps muscle, which is bringing my forearm bone closer to the proximity of my body. And that is brought about on this very subcellular microscopic level by the shortening of the sarcomere by the interaction of actin and myosin. Okay, so really fascinating stuff to see that the fact that these large kind of everyday actions are really just very small subcellular or, histological structures moving and bringing about these large impactful movements, right? So it's really fascinating to see that muscle is really a sequence of all of these sarcomeres doing their thing, shortening and bringing about such large important actions that we do every day. Okay. I'll take a brief pause here and see what questions we have. Okay. All right. Okay, what we're going to sort of introduce here in the remaining minutes is the sliding filament model. And the sliding filament model is what I've just been describing, how myosin is grabbing actin and dragging it towards the M line. And this is a model because no one has ever actually seen this. The physiologists who described this is a theory that they came up with. This has been polished and revised many times since then. Because of the nature of muscle, because it is excitable, and because we cannot actively see the histology of a muscle that is still alive, there's no way to do that just yet. They're kind of devising some ways with ultrasound technology and so on. But in order to see these structures on a muscle, we have to take a muscle out of the body, we have to cut it, freeze it, we have to prepare it, that frozen section, put it under a microscope, and that cannot happen with muscle that is alive. So this is a model or a theory. No one has ever actually seen this thing take place. We just look at these structures under the microscope and theorize that, hey, this is what it looks like dead. This is probably what it looks like when it's alive, okay? And so that is called the sliding filament model. And we'll talk about that, and we'll talk about cross-bridge cycling. So how those cross bridges, those heads of the myosin, stick up, make contact with the actin, and then drag it to the M line, and do that again and again and again and again in the cyclical process in order to generate force, okay? And then the other two things I mentioned we'll probably get into later on. So muscle contraction is really just a shortening of muscle, which we described previously, which is really just a shortening of some of the histological, the microscopic structures. And it is brought about by the thick and thin filaments that are overlapping, coming into contact with each other and dragging, the myosin specifically dragging actin toward the M line. Now, again, earlier physiologists used to think that these proteins shortened, right? That the actin was contracting or that myosin was contracting. Because the whole thing was shortening, they theorized that, hey, these things have to be contracting or shortening. In fact, we've come to realize that they're not shortening. It's not that they're becoming shorter, they're actually dragging. One of them is dragging the other, and so that's what's shortening the entire sarcomere. So they're kind of sliding past each other, right? And that is why we get that sliding filament model, that is, the name that this process is called. Okay, so within a sarcomere during contraction, there are a couple of things that change, and then there are some that stay the same. So because the area of overlap is happening between the A band, where we have actin and myosin, and because myosin is moving out towards the Z line, so here's myosin moving toward the Z line, here's actin moving towards the M line, because they're moving in opposite direction, and the A band is that area of overlap, the A band stays the same. It's like you're canceling out that movement, right? Actin is moving in towards the M line, myosin is moving out towards the Z line, and so the area of overlap is remaining the exact same, as we can see here. So here is the relaxed sarcomere. This is the area of overlap. Here is the shortened sarcomere, This is the area of overlap. And as we can see, the A band does not change because they're moving in opposite directions, because they're sliding past each other, and because A band is the area of overlap, those parameters stay the exact same. Other things that do change are the I band, the H zone, and the entire sarcomere, right? So the I band shortens. As myosin moves this way, the area of only actin gets shorter and shorter and shorter as myosin kind of takes up more of that space. Same thing happens in the opposite direction because actin is moving that way. The area of only actin gets shorter and shorter and shorter. Okay, and we can see that here. So here is the I band in the relaxed sarcomere, and here is the I band in the shortened sarcomere. The I band gets significantly shorter as actin moves that way and myosin moves that way. The H zone also shortens, as we can imagine. So the H zone is the area of only thick filaments, the area of only myosin. And so as these two things slide past each other, there's going to be more area where actin is overlapping myosin. And so the H zone in the relaxed sarcomere is this entire central region here, whereas the H zone in the contracted sarcomere becomes extremely small as actin is being dragged this way. It's going to take up more of that space for overlap. And so the area of only myosin becomes very small, becomes shorter. So the I band shortens, the H zone shortens, and then we see the Z lines coming closer and closer together. So the Z lines are way out here in the relaxed sarcomere, and as we drag these actin filaments closer to the H zone, closer to the M line, the Z lines become closer to each other. All right. Also important to note that this is happening because of cyclical formation and breaking of the cross bridge cycles. So we're not just reaching up and dragging it continuously. We're reaching up, dragging, letting go, reaching up, dragging, letting go, reaching up, dragging until we get that as close as it can possibly be. That's why we have this as a cyclical process, cross bridge cycling, because we're doing this in terms of forming a cross bridge, dragging, breaking that cross bridge, forming a cross bridge, dragging, breaking that cross bridge as we drag those out.